Welcome to this deep dive. Today we're exploring synthetic cannabinoids, and we're going to be looking at how they work on like a molecular level, how they interact with our bodies, and also the potential they might have for future therapies. We've got like a whole stack of research articles to go through.
It is such a rapidly evolving field with like a ton of really exciting research.
Absolutely, yeah. And our goal today is to kind of distill like the most crucial insights about synthetic cannabinoids for you.
Definitely. So why don't we kick things off by like setting the stage and talking about the endocannabinoid system, or the ECS. You can think of it as like this really complex communication network that exists in your body.
Yeah, the ECS, I've definitely heard of this before. It seems like it's involved in a lot of different functions in the body.
Oh yeah, for sure. It's involved in mood, appetite, pain, even immune function. The ECS has a hand in pretty much all of those processes, and the whole system is orchestrated by these cannabinoid receptors. The primary ones are CB1 and CB2, and these receptors, you can find them all throughout the body.
Gotcha. So these receptors, they're like the key players in the ECS.
Exactly. They're like these little signal receivers, constantly receiving and triggering responses to help keep everything running smoothly in your body. And what's really wild is that our bodies, they naturally produce these molecules that bind to these receptors.
Oh, really? So you're saying our bodies already have their own kind of cannabinoids?
That's right. They're called endocannabinoids. The most famous ones are anandamide and 2-AG. They act like messengers binding to CB1 and CD2 to cause all these different effects.
Wow. So in a way, our bodies are already doing this naturally.
Exactly. And the discovery of these endocannabinoids, this was back in the 90s by Dr. Machulam's lab. This discovery really kicked off the whole field of cannabinoid research.
It's amazing how one discovery can lead to a whole new field of science.
It really is. And what's even more interesting is that scientists now believe there could be even more endocannabinoids and receptors just waiting to be discovered. It's like we're only just scratching the surface of this whole system.
It's like this huge, intricate puzzle that we're slowly starting to put together. So where do synthetic cannabinoids fit into all of this? They're not naturally produced by our bodies, right?
Right. So synthetic cannabinoids, these are created in a lab to basically mimic the effects of those naturally occurring endocannabinoids.
Okay, so they're like these man-made versions of the molecules our bodies already make.
Exactly. They essentially act like keys that fit into the locks of the CB1 and CB2 receptors and trigger a response.
So they're mimicking what our bodies already do, but if they're synthetic, I'm assuming that doesn't mean they're exactly the same as the natural ones.
That's where things start to get really interesting. So scientists can actually tweak the chemical structure of these synthetic cannabinoids, which allows them to fine-tune their effects.
That's super interesting. So they can basically create molecules that are more targeted or maybe have a stronger effect on specific receptors.
That's the idea. And that's one of the things that makes synthetic cannabinoids so promising from a therapeutic perspective. It offers a whole new level of control and specificity that we wouldn't necessarily have with naturally occurring cannabinoids.
That's incredible. So we've got these receptors, CB1 and CB2, and scientists are essentially creating synthetic cannabinoids that can bind to them in different ways. But how do scientists even start to classify and understand these receptors in the first place?
Honestly, it's not a simple task.
Yeah.
So cannabinoid receptors are part of this larger family of receptors called G protein-coupled receptors, or GPCRs. And classifying these GPCRs can be tough because they all vary a lot in terms of their structure.
So it's more complicated than just looking at them and saying, oh, this one is CB1 and that one is CB2.
Yeah, it's a lot more nuanced than that. Traditionally, scientists would rely on the ligands that bound to these receptors to classify them, but that approach definitely has its limitations.
I see. So what's the solution then? How are scientists tackling this challenge now?
Well, they're starting to turn to more sophisticated computational techniques like machine learning. These algorithms can sift through mountains of data about the receptor structure and function to identify patterns and group them more accurately.
That's super cool. It's like using AI to decode these receptors.
Exactly. And this is really leading to a better understanding not just of CB1 and CB2, but of the entire family of GPCRs, which in turn opens up a whole world of possibilities for drug discovery.
It's amazing how technology is driving these breakthroughs in science. Let's go back to focusing on CB1 and CB2 specifically, and look at how synthetic cannabinoids actually interact with these receptors. I remember learning about something called receptor affinity, which sounded pretty important.
Yeah, receptor affinity is super important. It essentially describes how strongly a molecule binds to a receptor. You can think of it like a lock and key. Some keys fit better and turn that lock way more easily.
Okay, so I'm guessing a higher affinity means a stronger bond and therefore a more potent effect.
Yeah, exactly. And this is where those chemical tweaks we talked about earlier come into play. By modifying the structure of synthetic cannabinoids, Scientists can directly influence their affinity for CB1 or CB2. Essentially, they can dial their effects up or down.
That's fascinating. Can you give us some examples of how those chemical modifications actually impact receptor affinity?
Sure. So, one example is with these 1-purple-3 naphthoyl indole derivatives. Research has shown that just by adding a simple 2-methyl substitution to these molecules, that can significantly increase their affinity for the CB1 receptor.
So even small changes can have a big impact.
Totally. Another interesting trend is seen with these 3,2-substituted phenylacetyl indoles. These molecules generally have higher affinity for the CB1 receptor compared to their 3 substituted counterparts.
So it seems like the position of these substitutions really matters.
It's all about finding that right combination of chemical features to create a molecule that fits perfectly into that receptor's binding pocket. And then there's also this concept of stereochemistry to consider.
Okay, now we're getting into some pretty advanced territory.
Yeah.
What does stereochemistry have to do with all this?
Well, it turns out that the orientation of a molecule in space, its handedness basically, can also affect its activity. Dr. Meshulam and his team, they actually demonstrated that the psychotropic effects of cannabinoids are stereospecific.
So it's not just the chemical structure, but also the 3D arrangement of the atoms that matters.
Right. It's like trying to put a left-handed glove on your right hand. It just won't work the same way. And that level of detail is crucial for really understanding how synthetic cannabinoids are interacting with those receptors and how they ultimately produce their effects.
Wow. It's incredible how much detail there is at the molecular level.
Mm-hmm.
But we've talked a lot about the binding the affinity.
Mm-hmm.
What happens after that? How do these synthetic cannabinoids actually trigger a response once they bound to the receptor?
That's where things get really, really interesting. We have to delve into the world of signaling pathways, which is basically the cascade of events that happens inside the cell once that cannabinoid receptor is activated.
This is where things get really exciting. Lead the way. Okay, so we've got these synthetic cannabinoids. They're binding to the CB1 and CB2 receptors. It's like they're flipping a switch. But what happens next? What's going on inside the cell?
Well, it's like this chain reaction. Activating these receptors kicks off this cascade of events inside the cell, and these events are called signaling pathways. They're basically the way the cell receives and understands the message from the cannabinoid.
So it's not just a simple on or off switch. There's a whole process happening.
Exactly. And these signaling pathways, they can get really complex. There's a ton of players involved and all these intricate interactions. But let's break it down into a few key steps.
Right. Let's try to unpack it all. What would you say is the first step in this whole signaling cascade?
One of the main pathways involves these things called G proteins. They're kind of like intermediaries, relaying the signal from the receptor to other molecules inside the cell.
So the G protein is basically a messenger taking the message from the receptor to the rest of the cell.
That's a perfect way to think about it. And CB1 and CB2, they primarily interact with a specific type of G protein called GENIO. So when that synthetic cannabinoid binds to the receptor, it actually causes a change in this G protein.
What kind of change?
Well, it causes the G protein to release a molecule called GDP and bind to another molecule called GTP.
GDP and GTP. Those ring a bell from way back in biology class.
Yeah.
But remind me what they do again.
You can think of them like tiny little energy packets that control how active the G protein is. When GTP is bound, the G protein is activated and ready to pass on the message.
So cannabinoid binds, G protein gets powered up, and then what happens? Where does that message go?
Well, this activated G protein, it can then interact with all sorts of other proteins in the cell, which triggers a whole cascade of effects downstream.
Mm-hmm.
One of the main targets is this enzyme called adenylate cyclase.
Adenylate cyclase. I feel like I've come across that before. It has something to do with producing CANMP, right?
Yeah. CANMP is another super important signaling molecule. It acts as what we call a second messenger. It amplifies the signal from the receptor and triggers a bunch of different cellular responses.
So basically, by activating that G protein, the cannabinoid is ultimately causing a change in the levels of CAMP inside the cell.
You got it.
Huh?
And to be more specific, the GANIO protein actually inhibits adenylate cyclase. So that means it actually reduces how much CAMP is produced.
Interesting. So the binding of that synthetic cannabinoid can actually decrease the amount of CAMP in the cell. What are the implications of that?
Well, CAMP is a busy molecule. It's involved in all kinds of cellular processes. So messing with its levels can have a big impact on how the cell functions. For example, CAMP plays a role in regulating the activity of another key player called protein kinase A, or PKA.
PKA. So by changing CAMP levels, are synthetic cannabinoids basically controlling the activity of PKA indirectly?
You nailed it. And pKa is involved in a whole bunch of cellular processes, from metabolism to gene expression, so even small changes in CAMP can have this ripple effect throughout the whole cell.
It's like a domino effect, starting with that cannabinoid binding and eventually impacting tons of proteins and pathways.
Exactly, and it doesn't stop there. CB1 and CB2 can also influence other signaling pathways, adding even more complexity and diversity to their effects.
There's even more. Tell me about these other pathways.
Another important one involves this enzyme called phospholipase, or PLC. So activating CB1 and CB2 can trigger PLC. And PLC then takes this molecule called PIP2 and splits it into two important second messengers, disylglycerol, or DAG, and inositol triphosphate, or IP3.
DAG and IP3. Okay, I'm trying to keep
Yeah.
up with all these acronyms. So what do these molecules do in the signaling cascade?
They have different jobs, but they work together to regulate cellular processes. DAG stays in the cell membrane and activates another kinase called protein kinase C, or PKC.
Another kinase. So we have pKa and now we have PKC. There's a whole family of these signaling molecules.
They're super important for relaying and amplifying signals inside the cell. So while DAG is activating PKC IP3, it actually travels to the endoplasmic reticulum.
And just to remind myself, the endoplasmic reticulum, it's like the cell's calcium storage center.
Right. And IP3, it binds to receptors on the endoplasmic reticulum, which causes the release of calcium ions into the cytoplasm.
So what does that influx of calcium do?
Calcium, it's like a really powerful signaling molecule. It can affect everything from muscle contraction to neurotransmitter release, even cell death. It's like a big signal flare, letting the cell know that something important is going down.
So we've got G proteins, adenylate cyclase, pKa, PLC, DAG-IP3, and calcium. That's a lot to keep track of. But I'm starting to see how these pathways are all interconnected and how they each contribute to the overall effect of these synthetic cannabinoids.
Yeah, it's definitely a complex web,
Yeah.
but each player has its own role. And to really understand how synthetic cannabinoids work at the cellular level, we need to understand these pathways. But wait, there's more.
More? Really? What else is there?
There's another major group of signaling pathways that we need to talk about. They're called mitogen-activated protein kinase pathways, or MAPK pathways for short.
MEPK pathways. Okay, lay it on me. What do these pathways do?
They're basically involved in all kinds of important cellular processes, things like cell growth differentiation, even stress responses, you name it.
So they're basically controlling how the cell responds to its environment and to signals from within.
You got it. And there are three main MAPK pathways that are important to know: ERK, JNK, and P38. Each of these pathways has its own specific roles and targets, which adds another layer of complexity to the whole cannabinoid signaling cascade.
I'm starting to see a pattern here. The more we dig into these pathways, the more intricate and complex they become. It's like peeling back the layers of an onion.
It really is remarkable how much is happening at this tiny molecular level. And what's even cooler is that these pathways don't just act independently.
What do you mean?
There's a lot of what we call crosstalk between these pathways. They actually influence each other, creating this really complex web of interactions. And it's this web that ultimately determines how the cell responds to that cannabinoid signal.
So it's not just a linear cascade, it's more like a dynamic dance between these different pathways.
Precisely. And this interplay is super important for fine-tuning the cellular response and making sure the right signals get amplified or dampened depending on what's going on.
Man, I'm really starting to get a sense of just how incredibly complex this whole system is. It's like an elaborate symphony of molecular events with every player having a role in creating that final harmony.
That's such a great way to put it. And by understanding these signaling pathways, we can start to appreciate all the diverse effects of synthetic cannabinoids and how they might be used to help people therapeutically.
It feels like we've really laid the groundwork for understanding how these molecules work at the most fundamental level. Now let's switch gears and dive into the world of their therapeutic applications. It's pretty amazing to think about all these different potential uses for synthetic cannabinoids. I mean, we're talking about everything from nausea and pain to epilepsy and even neuroprotection. Where do we even begin?
Well, why don't we start with nausea? Because I know synthetic cannabinoids are already being used to help people who are going through chemotherapy.
Oh yeah, I've heard of that. It's really interesting.
There's this drug called nabilone. It's a synthetic cannabinoid, and it's actually already approved for treating chemotherapy-induced nausea and vomiting, which can be really tough for patients to deal with.
Yeah, I can imagine. It's great that there's something to help with those side effects.
Yeah.
What else are researchers looking at?
Well, beyond just managing nausea, there's a bunch of research into the potential for treating epilepsy. And studies show that endocannabinoids can have an impact on seizure activity, which suggests that there could be a therapeutic role for synthetic cannabinoids there.
So are you saying that we might one day see synthetic cannabinoids being used to help people manage seizures?
It's definitely a possibility. Of course, more research is needed, but the early findings look pretty promising. And then there's this whole area of neuroprotection, which is super interesting.
You're right, I remember reading about that. What exactly is neuroprotection?
It basically means protecting the brain from damage. And research is showing that certain cannabinoids have these anti-inflammatory and antioxidant properties. It's like they can shield brain cells from the damage caused by things like stroke or multiple sclerosis.
That's pretty amazing. It's like these tiny little bodyguards for our neurons.
Exactly. And we can't forget about pain management. Both natural and synthetic cannabinoids are known for their ability to reduce pain.
How do they do that?
Well, the research suggests that they can actually modulate pain pathways. So they're basically interfering with the way pain signals are transmitted in the body. It's almost like turning down the volume on pain.
It's fascinating how these molecules can have such diverse effects and so many potential benefits. But I have to ask, with all this promise, are there any challenges or downsides that we should be aware of?
Yeah, that's a great point. It's important to be aware of the potential risks, too. One of the challenges is the possibility of what we call off-target effects.
What does that mean?
It means that a synthetic cannabinoid that's designed to target, say, CB1, might also interact with other receptors in the body, which can lead to some unintended consequences.
This is like trying to hit one target but accidentally hitting another one?
Exactly. And then there's also the issue of tolerance, which means that over time, the body might become less sensitive to the effects of the synthetic cannabinoid, meaning you'd need a higher dose to get the same effect.
And what about the potential for abuse? I know some synthetic cannabinoids can have those mind-altering effects.
That's definitely a valid concern, and it's an area that researchers are actively looking into. They're trying to figure out what factors lead to abuse potential and develop strategies to minimize those risks. It's a complex issue, and we need more research to fully understand the long-term effects and potential dangers.
It's a reminder that even with all this potential, we still need to be cautious and do our research. But even with the challenges, the future for synthetic cannabinoids still seems pretty bright. What's next for this field?
Well, one thing that researchers are working on is designing synthetic cannabinoids that are even more selective. So they're basically trying to create molecules that bind really strongly to the desired receptor, like CB2, but have very little interaction with other receptors.
So like creating a key that fits perfectly into one specific lock and doesn't accidentally open any others.
Yeah, exactly. And they're using all these advanced computer modeling techniques to help them figure out the best chemical features for that kind of selectivity.
It's like solving a complex molecular puzzle.
That's a great way to think about it.
Yeah.
And it's not just about the shape either. They're also looking at how to fine-tune the way the synthetic cannabinoid interacts with the receptor once it's bound.
What do you mean? Isn't just binding enough to cause a response?
Well, it's actually more complicated than
Yeah.
that. Remember all those signaling pathways we talked about earlier? Well, it turns out that different ligands, even if they bind to the same receptor, can activate those pathways in different ways.
So two keys might fit the same lock, but one might turn it slightly differently than the other, leading to a different outcome.
Exactly. And that's the idea behind this really cool concept called biased agonism. So the goal is to create synthetic cannabinoids that activate certain beneficial signaling pathways, but avoid activating the ones that lead to unwanted side effects.
That sounds incredibly precise. It's like they're controlling the symphony at a molecular level, making sure only the right notes get played.
Exactly. It's like conducting the cellular orchestra with more precision and choosing which instruments to play and at what volume. And that approach has a ton of potential for developing new therapies with fewer side effects.
It's like personalized medicine at the molecular level.
You got it. Imagine a future where doctors can prescribe synthetic cannabinoids that are tailored to a patient's specific needs and genetic makeup to maximize the benefits and minimize the risks.
That's an exciting future to think about.
Yeah.
And with all this research going on, are there any specific areas that you're particularly excited about for the future of synthetic cannabinoids?
One area that I'm particularly interested in is the potential for treating neurodegenerative diseases like Alzheimer's and Parkinson's. There's increasing evidence that the endocannabinoid system is involved in these diseases, and it seems like synthetic cannabinoids could be a new way to try to treat them.
So they could potentially help protect the brain from the damage caused by these diseases.
That's the hope. The early studies in animals are looking pretty promising, but of course we need more research to see if those findings translate to humans and to make sure any new treatments are safe and effective.
It sounds like there's still a lot to learn, but the potential is definitely huge. Are there any other areas that have you excited?
Another one is the development of synthetic cannabinoids for treating chronic pain. A lot of the traditional pain medications out there have all of these side effects, and a lot of people don't actually get enough relief from them. So synthetic cannabinoids could offer a real alternative.
So it's about finding better ways to manage pain and improve people's quality of life.
Exactly. And the research in this area is moving really quickly. We're seeing new molecules being developed, clinical trials are starting, and our understanding of how the endocannabinoid system is involved in pain is growing all the time.
It's amazing to think about how far we've come in our understanding of cannabinoids. We've gone from just knowing about cannabis as a plant to
Well,
actually understanding its individual components and how we can use them in medicine.
it's a real testament to the power of science and how curious we are as humans.
Uh,
We've gone from this stigma around cannabis to exploring the intricate world of these molecules, receptors, and pathways.
and we've seen that science unfold right before our eyes.
And with all this new scientific knowledge, we now have the ability to design and create targeted therapies that could really change the way we treat a whole range of diseases and conditions. From managing nausea and pain to potentially treating these neurodegenerative diseases, the future of synthetic cannabinoids is really exciting.
It's like we've opened this door to a whole new world of possibilities.
And I can't wait to see what amazing discoveries come out of this field in the future.
This has been a really fascinating deep dive. I've learned so much about synthetic cannabinoids and the potential they hold. Thank you for sharing your expertise with us.
It's been a pleasure to talk about this with you and with our listeners. I hope you've all learned something new about the incredible potential of synthetic cannabinoids and how science can help us understand and improve the world.
To our listeners out there, we encourage you to keep learning and keep asking questions. The world of science is full of amazing discoveries, and we hope you'll join us next time for another deep dive. Until then, keep exploring.